Hello, everybody. My name is Michael Black, and welcome to our podcast, Excipient Talk, brought to you by galenIQ. And today I'm talking to Annette. Hello, Annette.
Hello, Michael. Nice to meet you.
Nice to see you again, too. So, Annette, please introduce yourself to our viewers.
Okay, first of all, thank you for the invitation. My name is Annette. I'm with GLAD for over 20 years now, so quite a while. I'm pharmacist by training and did my PhD in pharmaceutical technology. I started here at GLAD as a project manager in pharmaceutical development, which gave me a good foundation in this field. For 15 years, I led the pharmaceutical development department, and since the beginning of last year, I've moved into a staff role as a scientific expert. These days, I focus on collaborations with universities, supervising or mentoring master students, keeping the eye on new technologies and anything else that looks promising or just plain interesting for us. And I really enjoy connecting science with innovation, and I'm always curious about what's coming next.
Okay, yeah, sounds great. So, what are we going to talk about today?
So we recently presented a poster at the 5th European Conference on Pharmaceutics in Porto. And one of our strengths is improving the solubility of poorly soluble substances. And this time our new approach was to use nanoparticle-based formulations in our standard fluidized bed equipment. And by reducing the particle size of active pharmaceutical ingredients down to the nanometer range, we can significantly increase the surface area of contact between the active and the medium. And this helps enhancing the dissolution rate, the cell saturation, solubility, and even the interaction with cell membranes. And all of this contributes to better bioavailability.
So thank you very much for that explanation. So your poster also had a very intriguing title, which was Nanoparticles Meet Fluid Bed. And you've explained a little bit of the theory behind it. So what did you actually do in the lab?
So in fluid bed processing, we tested two things. We had drug layering and granulation, specifically for loading nanoparticle formulations. We used different carrier materials. For drug layering, we used phthalates, which are microcrystalline cellulose starter beads, as insoluble material they were bought from Ingredient Farm, and as well as isomalt particles as soluble carrier, which we got from Beneo. Thank you for this. For granulation, we used mixtures of microcrystalline cellulose as well as isomalt with a smaller particle size distribution. And the main goal was to see if we could keep the properties of the nanoparticles, like their size and stability, while turning them into solid dosage forms. And so we really focused on making sure that the nanoparticles stayed intact and effective through the entire process.
Okay, so that was your goal. And what did your results show?
The particles which we got out of the process were compact and had a nice and even size, which tells us that both processes, the pellets drug layering and the granulation processes, worked quite well. We also managed to load more than 10% of the active as nanoparticles into both forms. And if needed, we could even go up to something around 40%. And I think we could go higher as well. We only tried 36. This was easily possible. When we checked the redispersibility, the nanoparticles stayed pretty much the same size as the original nano suspension. The increase in average size was less than 25 nanometers, and the polydispersity index, the PDI value, stayed low, which is a good sign for their stability. And finally, the in vitro dissolution test showed a clear improvement in how fast the drug dissolves, both for the granules and the pellets compared to the raw unmilled API.
Okay, well, it sounds like a very successful study. And how was the reaction to your poster when you showed it in Porto?
The reaction for the poster was very positive. Many attendees were interested in the approach, especially the combination of nanoparticle technology with fluid bed processing. The people appreciated the practical angle of improving the solubility and the bioavailability, something that is still a real challenge in the formulation development. And there were some great discussions around the redispersibility results and the potential for scaling up this process. And overall, the feedback confirmed that this strategy is not only innovative, but also highly relevant for current pharmaceutical challenges.
Okay, great. So what could be the next research and development steps from your side?
So we showed that the fluid bed granulation and the drug layering work really well as follow-up steps for making nanoparticle-based oral drug formulations. And by combining the wet ball milling with the fluid bed processing, we were able to get consistent particle sizes, solid and compact products with a good redispersibility and much faster dissolution rates, which we expected and wanted to show. And overall, this method gives us a smart way to deal with poorly soluble drugs by making them dissolve better and more available to the body. So the next step would be to turn our pellets and granules into actual oral dosage forms, such as capsules or tablets. That's where it starts to get really practical and applicable to the patients. And we are also planning to apply this method to other poorly soluble APIs. The idea is to build here a broader data set and to see how well the approach holds up across different compounds. And it's all about learning more and seeing what's possible with these technologies and these excipients.
Yeah, and it sounds very exciting because, as we know, this is a very important and critical issue in the pharmaceutical industry at the moment. Poorly water-soluble drugs and bioavailability. So it sounds very exciting. I hope in your future work that galenIQ also plays a strong role, and we're there to support you as much as we can. And yeah, thank you very much for joining today, and I wish you every success with your further studies in this area. And I hope to see you again soon. And thank you very much. So everybody, thank you for joining today in our podcast Excipient Talk, brought to you by galenIQ, and we will see you again soon.
Thank you very much.